All right, let me explain something to you. They're talking about putting some DNA into you that is not your DNA, and you shouldn't do that. And it came from aborted fetuses or something. They coming up with all kinds of things. Let me tell you what. Every single day, get all kinds of bacteria into my bloodstream. I have all kinds of cuts and crushed fingers and everything. See, the black is finally worn off of my fingernail and it's starting to regrow. But you do too. All you have to do is puncture your skin and get a little tiny cut. Instantaneously, you have some new DNA has entered into your bloodstream. Your body has to come out and kill that invader. It's literally an invasion. And your body is equipped to do that if you have the correct bacteria and enzymes inside of you to attack the bacteria and enzymes that are coming in to attack you. It's a fight. All right, this is the heart of the matter right here. That is that fluid-filled highway. I call it a tubular-filled highway. Now, just below that is your vital flesh. Above that is skin, which is, it's made to be just abraded and moved to go away. And say your fascia that coats your lung. Let's say this was a lung, and you had your fascia coating it. I'm sorry, your interstitium. And this right here is the source of the membrane. You're going to have some gooey stuff here that are cells that can move and slide and do this and that. Those are connective tissues. Those are connective. They connect and they slide and they move around so that you can do all that kind of stuff and they don't come apart. But right below that is the fluid-filled highway. Now, if something breaks through this mat that's supposed to be here, that's not supposed to open up, and you get a cut. Let's just say you get a cut. Well, coming through that cut is going to be mud and dirt and some nasty, who the hell knows what's coming through. And it's going to get into you. And right here, your troops are going to come in and wipe that stuff right out. That's what it's made for. And that is what's called enzymes. And this is literally your immune system. And every single organ has that same layer. Every single muscle, tendon, lymph nodes, everything, all your organs, your intestines. That's why there's 200 types of cancers. Every one of them is a different type of invasion through a different type of molecular opposition. Something there is fighting back, and down here might be acids or salts or something, or who knows what's going to be down in this particular organ. But some type of an invader says, ooh, I like that stuff down there. There's all kinds of copper down there. Oh man, there's some, who knows what over there, silicates. We got to get down there and get some of that. See if you can find a hole. And they start digging around. And your guys should see them. Say, whoa, there's a guy digging around over here. Go get him. See you later, alligator. And they never get there. However, if you don't have a guy sniffing, first of all, you got to have the sniffer. I'm going to show you this. They've already found this is true. If you don't have the sniffer, you never see them. Then they start digging away until they can find a hole. And they'll find one sooner or later, and in it comes. That's a predisposition to invasion because you don't have any troops to protect you. And that's the problem with these little bitty kids. They don't have any invaders. I mean, any immune response. So you get them with a live vaccine, even though it's just very, very barely alive, it can find its way in there and take root, and the kid can get extremely sick. That's just a fact of nature. If you don't have anything to protect you in that layer, the solution to this is to know what in this layer protects you from this invasion. And the only way to do that is to get a database of what is in the gut microbiome, which creates that layer. That's created by your gut microbiome. That's where bacteria are propagated. That's where they live. That's where they thrive. That's where they engage with everything that comes through your body. And everything that comes through your body, you're going to have all kinds of foreign DNA going into you every second of the day, basically. DNA is everywhere. It's not a big deal. Your body knows how to deal with it. Even if it pokes into here, that's okay. Your body should be able to deal with it. That's what immunities are, basically. If you have nobody to come and help you with that invasion, well, then you're in trouble. And that's what they never even, this is just discovered. Well, they just agreed, yes, it is true what I've been saying. This is a layer and this is what it does. And then they did some looking at this layer, and wait till I show you what they found. It says your textbooks are wrong. All right, this goes back to 2018. They just said, yes, this is the biggest organ in your body, a new organ we never knew was here. And the reason was it was flat as a pancake and there was no fluids in there when they did the autopsies. They thought it was just one sheet. So that's that. All right, my friends, I just saw Kennedy talking about mumps, measles, and rubella vaccine, and then talking about these cell lines coming from aborted fetuses and so forth. Let's talk about this, because we need to get some really factual information here. And if I'm wrong, I want to be told I'm wrong by somebody that has an ear to the president or to Kennedy. All right, check this out. That's what I showed you. That's that fluid-filled highway. Now this is a shot of it this is like the skin up here. Then you got the fluid-filled highway. This is the thing that protects you. That's in between your skin and your vital flesh. You have the layer of membrane down here, which is the layer that the bacteria grow in, and they collect up these little ribosomes or whatever these are, some kind of enzyme, but they're ribosomes, and they squirt them out into these little channels, into the fluid-filled highway. And at that point, they go wherever they have to go and protect you. Now, we can actually see this happening. Watch. All right, so now this goes back 5 years ago. This is Gokal. He's with UC Berkeley, and they're looking at this interstitium layer in the real time and how fast this works and the components to make it work. And as you recall, I showed you that picture, that real colorful one with the fluid-filled highway and the layer below. He's going to be showing this. Now watch what he shows. I'm going to stop it here and there probably. Here goes. The name of this is Your Textbooks Are Wrong. This is what cells actually look like. All right, so here we go. Here goes Gokal.
Something that I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is basically within each cell.
This is an unprecedented view.
All right. Remember that area I showed you before? It was down here. I think it was blue and green and stuff. These are the fluid-filled highway entrances, basically on and off ramps to get into that fluid-filled highway. Up there is the skin. Remember all that skinny looking stuff? Now watch. He says this is the view of the cellular world where we can actually see immune cells scooping up sugars. Now they say sugars, I don't know about that. They could be, but they could also be enzymes and ribosomes. Here goes.
View of the cellular world where we can
All right, you see that? That's one of these guys running around grabbing up all this blue stuff, and they think it's eating it up as sugar. And it might be. I can't say it isn't, but I know what it is doing. It's gathering up something in here, and it's bringing it out into the fluid-filled highway through these on-off ramps.
actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
See that? There it goes.
Focusing only on the crawling immune cells, we've noticed two classes of them.
Two classes. One of them here is a sniffer. That one there goes right walking around. Whoops. And that one here is just traveling around in the body. Hold on one second. Let me grab a hold of a representative little thing here. Now, this is what the sniffers do. You see over here? It's going to go through here, and it's got a whole batch of little magnetic signatures on it. And if that matches something that it doesn't like, it runs back and tells the guys that are built like this, which are the ribosomes. And it says, go out and attack that guy. So here's what Gokal is going to say. Two classes of bacteria, of these immune cells. it seems, was not hungry at all, this one. It doesn't eat anything. It just doesn't gobble anything. It just goes around with this little magnetic signature looking for something that matches. It was very active. It moved around a lot, but it didn't eat anything.
One seemed was not hungry at all, but it was very active in terms of trying to figure out, what the environment is.
See, trying to figure out what the environment's going around sniffing. Hey, anybody out there I don't like? Any bad guys? You see, here it is right here. All right, and if it sees one, it goes back and tells the big boys.
There's another one that was kind of...
That's right there. So it either comes back or somehow it triggers that guy to say, hey, we got a trouble out here. We got a spectroscopic nastiness. Come out here and grab some of this, whatever it is, and whack them with it, and it does. And then it did. And that happens in that interstitial layer.
sloshing around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment.
Now that's what we need to do, is to find out what are these things attacking. When you get hit with these MMR vaccines, what does it do to your immune system? Your immune system is going to respond. That's what it's all about. I don't care if it responds in a positive way or a negative way. At this point, there's a response. That is the case closed. There is no argument about that from anyone, I would hope. If you're going to hit something with something you want to cause an immune response, you're going to get one. Now, is it just a little bit enough to alter the chemistry so you will never have to worry about it again because you've created that sniffer? Now that sniffer's going around looking and there's somebody he can report to? It's a sequence of everything. It's like you need the right guys at the right place at the right time. So you got your sniffer. If that thing's done, you're done. Then you got your thing that runs around grabbing all the stuff, but you got to have the stuff to grab. And then it goes out and delivers it. Now let's actually watch that happen. Well, we just didn't see it happen, but let's look at it in a different way. Okay, so let's just have a little discussion about this. I showed you Go Call and all of that stuff being harvested up, going up into the fluid-filled highway, which was just announced that it is a newfound organ and the biggest organ. Well, they're still debating if it's the biggest organ in the human body, but it obviously is. It covers every single thing in the body, including red blood cells and everything. Everything has a membrane, 100%, and all membranes are made from phospholipids. All right, so you have to understand chemistry, biology, anatomy, all that stuff. Now, what is floating in that fluid-filled highway? What kind of cars do we have? Well, those cars are enzymes. They're bacteria and they're enzymes. And I showed you, as they come out into the fluid-filled highway, they take off. I say that thing that was scooting around in here was a bacteria harvesting enzymes. Let's think about it in that respect. Because enzymes are what are required to do the chemistry. If you don't have the enzymes, you're going to die. You're dead. Seriously, that fast. Okay, this is really quite simple. We have 75,000 or more enzymes in our body, and bacteria create the enzymes. Enzymes come out in a little ball. It's called a ribosome. So we have literally, 75,000 or more different bacterias roaming inside of our body, creating these ribosomes. Every one of these is a chemistry set, and I mean elegant. I'm going to show you this in very, very extreme detail. Now, they're talking about getting somebody else's DNA in you and this and that. Well, everything has DNA. There's a program, which is the DNA. That's the program that runs this bacteria. It says, I'm going to have these little things sticking out. I'm going to have this flagella. I'm going to have whatever it is. And then on top of that, in that DNA, it says, I'm going to make ribosomes. That's my job in life, to make ribosomes. And it squirts them out in these little balls. Blip, 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 blip. And they just float around. They don't eat. They don't poop. They don't do anything until they get triggered. And what happens when they get triggered? Well, let's talk about that. All right, let's walk through the stages here. We know we have the interstitium, which is the fluid-filled highway. We know we had something going on in here which was squirting out through these little on-off ramps into that fluid-filled highway. Quite obviously there was some critter running around gobbling up something, squirting it out there, which was one of these, almost certainly, which is a bacteria. And they have, some of them do and some of them don't have flagellas. A flagella just goes zip and it takes off going down that highway a million miles an hour. So we know this is happening. What actually is it that happens when this thing, which is a bacteria, and we have thousands and thousands and thousands of different species, what does it make when it makes a ribosome? What the hell is a ribosome? And where does all this stuff happen? Well, first of all, it happens in the gut. In almost all cases. They call it the gut-brain axis. Your brain and your gut work together. And what happens is when you have the right bacteria, beneficial bacteria in you, in the right quantities, they keep the harmful bacteria under check. These things are in us. Almost everybody has all the bad stuff. But if they have the good stuff, the bad stuff cannot take over. All right, now, this is talking about fecal matter transplant, which should be renamed to be called digestive enzyme replacement, bacterial replacement. Because what you need to do is get the beneficial bacteria back into somebody that doesn't have it. This is where it grows. It's not going to just come in here and start growing by itself. You've got to replace it somehow. And that's why they say eat this and eat that. And there's cultures on earth that do all kinds of crazy things and eat all kinds of crazy things, but they get healthy because we think it's bad for you. And some of this stuff is, no question, but some of it is good that has never been thought about before. And they're enzymes. Enzymes are just a fact of all life has enzymes in them. 100% of life has catalase. Catalase is an enzyme. 100% of life. So enzymes are ubiquitous. They are everywhere and they do everything there is done in life because they work so fast. And that's what an enzyme is the ribosome. So let's look at what this ribosome actually looks like. There's the bacteria we're just talking about. There's the DNA. It has its own DNA, and it just says, I'm this bacteria, I'm going to have this, and a flagella, I'm going to do all this stuff, and I'm going to make this exact sequence of proteins. All right, there's 20 different amino acid proteins. They're going to come out in that exact sequence. Exact. Every single time, 100% of the time, and they are going to program exactly that sequence. Exactly. Or it's genetically wrong. And that comes in just like a typewriter into the transfer RNA, which is your messenger RNA. It goes transferase, is this cloudy stuff, and it goes and these are the ACTGs that come along here, and they add a uracil, the U, that makes it, I don't know, the U splices into the DNA somehow using another enzyme, RNA polymerase. And that clips right into our DNA, and then you now have this new program. Now, is it a good program or is it a bad program? That's the key. The protein chain, if it's wrong, it's wrong. So it goes in here and it's wrong. What it does in there, it's wrong, I don't know. It may give you an extra finger. It may do whatever it does. But unless that protein chain is exactly in the sequence it has to be, the outcome is not going to be good. Or it may be better. I don't know. That's the key. That's what happens in DNA as it changes and evolves and you become immune. And I believe this is even memory and everything else happens in there because the DNA is so sophisticated. 98% of it, they say, is just junk. That's ridiculous. You think of the complexity of the human body is just unbelievable. And it only uses 2% of the DNA to make that? It's nonsense. 98% is your memory, your immunities, your all this different. And twins, one of them goes this way and one of them goes that way. Identical twins. So there's not the same DNA for the rest of their life. The junk DNA goes like this to this guy and like that to this guy, whatever it is. Whatever they run into in their life has to affect their DNA. To my way of thinking, DNA is just a gigantic memory stick. Okay, everybody knows what DNA is. It's that double helix that wraps around a coil like this. Now, there's 4 different proteins involved here, CTAGs. All right, CTAGs. You see them here? And they can only, an A can go to a T either way, but it can't go to these. The C goes to the G goes to the C, and so forth. And they're not very complex little chemicals, but they are specific, and you know, they didn't just happen by accident. Now, you see all these phosphate groups, phosphates, all those P's are phosphates. There's phosphates everywhere. The phosphates here is phosphorus and sulfur and silicon. Right in this area is where your membranes are constructed. So at any rate, there is, I believe they claim now there's 3.2 billion base pairs in the human DNA spiral coil here, the helix. 3.2 billion. Now, I had DNA tests done on my stuff. And here's what they are, the CTAGs. And this was from a sample, 36-inch tip, which, here's the different sequences. Now, if these sequences match precisely the sequences in the mitochondrial human bacterial DNA, which they did, it's 100% match, there's no question whatsoever, this was from a Homo sapien well, here it is back here. Homo sapien mitochondrial cytochrome B and D loop region. And it said it was excellent quality DNA. That's the stuff I had tested. So this is not a joke. This is not silly, the stuff I'm showing about these body parts were literally living creatures at one time, and the exceptional preservation is exceptional. Literal blood. You know, I harvested the samples myself, sent them off to the lab, they tested them. He says he'll stand behind the results, but you have to know it was me, Roger Spur, not Helix Biolabs, that drilled into these specimens. I drilled into them, took the DNA out very carefully, and it was dense and heavy and excellent quality. Excellent quality DNA sequence from both of this, 36-cent and the lung. I mean, and it's just, you can't miss it. There's no way to dismiss what it is. Here it is right here. Hold on a second. Give me 1 second. There's the lung. You drill down inside of where there's blood, and it's just saturated with blood here. At this tip of this lung right here, there's just blood everywhere. And this was all from the Great Flood, and they died flat on one side. All right, even the heart was still here. That's where the heart would have been. Died laying just like that, flat on his back. And all that stuff boiled away. And the fascia, this is heavy pleura. Pleura is like 10 times or maybe 100 times denser than actual fascia. This bone has fascia, which is a coating, it's a wrapping, and they used to call it tunica. You can actually see it coming here. You see that coming here and coming here? It wraps right around like a crescent roll and seals right along these borders. So it's in its own, they called it tunica, the ancients. Of course, that's the broken part. That's the onlyest little bit of bone left. That little tiny, tiny bit. If you can see that white spot. And in the microscope, you can see where it transitions. All of this is called nucleophilic invasion substitution. It's very simple once you understand it. Meat transitioned completely exactly like it was a piece of meat. And there's the fascia that comes down. This is what I studied in detail. How thin it can be, and then how heavy it gets right where it seals and connects to the next body part. And that right there is one of those spots. There's a little latch right there. And that has to connect, and there's two little dots at the end which drain the fluid from here into the next organ. All right, that's why we have so many cancers. Every organ has its own set of chemistry. And every set of chemistry requires an enzyme. Let's look back at the enzymes. All right, you see this right here? Look at this. This is about living organisms and biosynthesis. Well, what's biosynthesis mean? Bio is biology. Synthesis means synthesizing something, making something from bits and pieces, synthetically making something, biosynthesis. Well, what does it have to do with what we're talking about? We're talking about amino acids. And they are used to construct things in your body. It's extremely important for virtually everything in your body. And they really don't know much about them, to be perfectly honest. They know they're there. They know there's 20 of them, supposedly, at least. But how they got there, nobody knows. It says some amino acids can form naturally under the right conditions. This is important for theories about the origin of life. Now, they simulated, this Miller experiment in 1953 simulated early Earth conditions using water, methane, ammonia, hydrogen, lighting, and successfully created several amino acids. Well, maybe. In cells, using genetic codes, cells don't create amino acids during protein synthesis, but they use amino acids from where? To build proteins according to instructions encoded in the DNA. Where did they come from in the first place? The transfer RNA molecules carry amino acids to the ribosomes, which we have talked about or will, where they are linked into chains, which is the polypeptide chains, and then they transfer those chain programs into messenger RNA. That goes to your DNA and changes your DNA. Okay, let's just put it in a nice little package with a ribbon around it. That is the ribosome, which this particular type of bacteria, this species, creates. That ribosome just squirts out of here. When it squirts out, it goes into the human body as a ball. All right, and that ball is loaded with this program of amino acids in an exact chain. Some of them are short chain. Some of them are very, very long. It depends on the job it's got to do. And in that ribosome, it programs messenger RNA, which is the stuff that goes out and invades the DNA. All right, so the bacteria creates the ribosome, which is nothing but a big ball of these little things turned into a little tiny ball. All right, and that ball is now floating around throughout your system. And there's a billion, you can fit 1 million of these, they're so small, into one human cell. 1 million. The things that I'm talking about, people their minds can't accept them because it's just too, the numbers and the sizes, and it's just amazing. But anyway, these, when they're called upon, they trigger. And how that happens, I don't know. Something might come up and go, click, and something pops off like this and it goes, but the end result will be an extremely elegant molecule that will be exactly identical to this every single time. Every one of these colors will be in the right order, the right distance from each other. They will have the correct dipole molecular moments, which means there's going to be areas of magnetism and non-magnetism and positive and negative, and then they create atomic click chemistry. I looked it up. There was a guy in 2001 or something that came up with the term click chemistry, which just means something that happens predictively, and it's basically the same thing. So mine's about enzymes. So anyway, they will create a certain pattern, and here's that pattern. I didn't want to steal the guy's name, click chemistry, but here's what happens. That's going to be exactly the enzyme, exactly identical. It's not going to be able to, if that's pointing that direction, it's not going to work. Or it might work just barely, or it might, I don't know exactly what it'll do, but it's not going to be right. You have to have the identical same enzyme every time to do the job. And if it's damaged, or it's not there, or it's genetically been changed, which is the case of plant modifications and so forth, they change them so that they don't react to things they normally would react to. That's what they do. And all you got to do is go click, click, click to the right things that are doing that good job, and turn them off. Or you could. My point is, you can change the outcome of these just by making a tiny little click, or clipping a piece out and putting a piece in, and it makes it a whole different animal, basically. Because that's what this does. It's click chemistry, and it doesn't get used up. It goes and catalyzes all of these chemical reactions to do literally up to billions of years of chemistry in less than 1 second. And they don't even get used up. They just keep going on, and they're machine gunning everybody through that molecular dipole magnetic moment signature. This is all dipole electron flood theory. It's just everything's a dipole. Done. So we know we got the bacteria. We know the bacteria make the ribosomes. We know the ribosomes create the messenger RNA sequences. We know the messenger RNA gets into the DNA. We know that it requires an enzyme for every single step of this process, every single step. Now, what are enzymes? What are they? Remember I talked to you about the little ball? That's the ribosome. When it gets triggered, the sheath goes, out comes some kind of gigantic ball of particles. All right, now, are enzymes proteins? Well, they are and they aren't. All right, well, what does a protein do? A protein is a building block. A protein builds things. Proteins build muscle and bones and all kinds of things. Well, what does an enzyme do? An enzyme does exactly the opposite. It breaks things. It kills things. It breaks down food into its tiniest little particles. It does half-life chemistry. This goes the other direction. This takes those products that this breaks down, done, and it says, okay, I need to give me 6 of those and 12 of these and 15 of these, and it puts them together, and it makes a piece of muscle, whatever it is. This is like Amazon. It's just like the construction industry. All right, this is a paper I put up on academia.edu because they won't let you publish unless you're peer reviewed. Nobody would peer review it. So this was about fascia-facilitated fossilization, creating my mud fossils like this goose. And there's the feathers in his head and everything. This was, during this research of these fossils, I discovered a layer that nobody had ever seen before. And this is just the abstract. You can get the full paper up here anyway. If not for fascia, you would be a bag of gushy mixed organic molecules, metal molecules, dissolve in the rain. You just wash into nothing. Fascia is the coating and the reason you stay together. It is the strings and springs in connective tissues. Fascia is only recognized as important in the last few years. This goes back to 2015. Nobody even ever looked at fascia as anything other than just packing material, and they didn't know about the fluid-filled highway. So here's the key takeaways from my paper in 2015. Now, I've been studying this for over 10 years. Fascia protects every bit of you from invasion, including a complete fabric bag that is your skin. And we're going to look at this in a second in great detail. Mudfossils contain all body parts, and each is the product of fascia-facilitated fossilization. They coat organs, they coat muscles, they coat bones, like this. That is fascia that coats that. This is the same, it's collagen. But inside, it's no more collagen. Collagen is made basically of membranes, and membranes are made of phosphorus, phospholipids, and when they boil down into aluminum silicates. So I knew what I was dealing with was a layer that was unknown. So they contain everything. To me, this is my suspicion. It is now suspected fascia connective tissue may be fiber optic, because it goes to every single cell in your whole body. It goes everywhere. Fascia is a tubular, fluid-filled fabric network. They call it a fluid-filled highway now. Instantly reconfigures itself as damaged or pulled apart. You go through your fascia and cut through. That's cutting through your membrane. That's membrane. Every one of those is a membrane. It's got to come right back together and seal itself up. Otherwise, you get invaded. This is what I've been studying for a long time here now. As a component of support and posture, fascia is also suspected to be a source of many pains. Yes, it is. I can guarantee you that. You have to very, very gently, slowly pull your fascia apart. When I first started in this, I got a hold of the guy that did, it was called the fuzz speech, was about fascia. They called it fuzz. It was just there as like padding. And he said, yes, it's obviously is the source of a lot of pain. And he said he knew when people were like stuck almost. Because when this was a guy that does autopsies, he said when he would cut into them, he'd have to actually almost couldn't cut through some of the fascia. It was so bound up and tight and never had been stretched. You just don't move, and all of a sudden nothing moves anymore. And trust me, that I know this for a fact. If you don't lift your legs up basically every day, just nice and slowly, and pull them up and get up in the air with things, and slide your back over this way and that way, and you're going to eventually lock up. That's what walking basically is all about. And do a little bit of just very gently, not to the source of pain. And I don't give any medical advice here. This is just, everybody knows you have to stretch and do that. That's all I'm saying. All right, when I started this video, I said Robert Kennedy needs to see the new evidence about the interstitial layer, this fluid-filled highway, how it reacts with the enzymes, and how the enzymes are affected by bacteria, and how the vaccinations and so forth can affect a kid that has no immunities whatsoever. I think I've been pretty clear about what I presented. I'm going to leave it at that for today, but I would love to engage, and I just was contacted by Marguerite. I don't know if I showed you this or not, but she's over at The Hague in the Netherlands giving a presentation to a bunch of people about autism and the gut health, which affects everybody in the world. Nobody's immune from that. It's not just like some here and some there. Everybody now. Everybody has issues with their guts. And why? We think we have some answers to that, and hopefully we'll have a chance to speak about it. But these are the kind of people we need to speak with. So anyway, I'm going to leave it at that. Thank you. I love you all. And Marguerite, you're doing a fabulous job. Excellent presentation. All right. I love you all. Bye.